VC Chemistry, Unit 4, Hydrolysis of Protein. So in this session, our major goal is to be able to distinguish the hydrolysis of protein versus the denaturation process of protein. Okay. So hydrolysis, we talked about before, when amino acids come to form polypeptides and therefore become a protein, the process is called condensation reaction, right? So hydrolysis is the reverse of condensation reaction. So when a polypeptide, basically protein in food, goes into our body, our digestive system will catalyze or have different enzymes to catalyze the hydrolysis reaction to add water back on to those amino acids. So our digestive system breaks protein down to amino acids. And when it's transported in the bloodstream, when it reaches the target location, then the synthesizing process starts. So again, those amino acids, through condensation reactions, form the protein that's actually needed by the body. Okay? So here is a picture of a hydrolysis reaction. So you see that there is an amide linkage, or you can say it's a peptide linkage in the polypeptide chain, right? Hydrolysis is breaking that carbon-nitrogen covalent bond so as to add the OH back to the carboxylic carboxyl group of one peptide, and then adding the H to the another maybe peptide or sometimes amino acid. Okay, so you're just adding water back on. Okay. So peptide bonds are broken in hydrolysis. Water is added to the peptides, and basically this entire process forms shorter and shorter peptide chains until they're all amino acids. Now, before we talk about denaturation, so how are these two processes differ from each other? Okay? For denaturation, we know that it is actually the bonds in tertiary structure that are broken. So for sometimes can be remade in a random way. So for example, all those hydrogen bonds or ionic interactions among the side chains on the different parts of the polypeptide chain, that they are responsible for tertiary structure. And a lot of time it's due to either extreme temperature or extreme pH. Those hydrogen bonds are disrupted, or those ionic interactions are disrupted. So as a result, the tertiary structure of a protein is destroyed. That's denaturation. However, the molecular formula of that protein remains the same. So in the process of denaturation, you're basically breaking down either the tertiary structure or sometimes the secondary structure. But your primary structure remains intact. That means that polypeptide chain remains intact. Okay? So the molecular formula is exactly the same before or after denaturation. The protein is no longer useful, or you can say that if the protein is acting as an enzyme, then the enzymes gets totally destroyed because the shape of the active site gets totally destroyed. But the protein itself, like the primary structure, the polypeptide chain, the sequence of amino acids remains intact because that covalent bond that forms the amide linkage remains the same. Like that cannot be changed just by changing temperature or pH. That's just not strong enough. Okay? But in hydrolysis, the covalent bonds holding the primary structure are actually broken. Okay? And water is added to the molecule. So the entire polypeptide chain is broken down into amino acids. So the molecular formula changes, and as a result, the peptide chain is gone. The polypeptide is gone. It actually turns into their substitute units, which is basically amino acids. So you can see that hydrolysis and the denaturation, they both destroy protein, but they destroy protein to a different extent. So you can think about, like, for example, whenever we cook egg, right, we are actually, when we cook egg, what happens is it's not really hydrolysis when we cook the egg, right? The process that happens when an egg is being cooked is denaturation because we're only destroying the tertiary structure, right? So whatever enzymes are in that egg is no longer working. That's what we actually want, because we don't want to eat those weird like micro substance that are actually in the protein. We want to destroy them, but through the process of cooking, the primary structure remains intact. That's why when we eat that cooked egg, our body are still able to utilize the amino acids that are actually in those protein to be useful for our body. Okay, but when we eat that egg, this is when hydrolysis happens. The proteins, although they're denatured, but the primary structure is intact. But when we eat it, this is where covalent bonds gets broken by the enzymes in our body to catalyze that hydrolysis reaction. And then the resulting product is amino acids that gets transported into our body. Okay? So if it's hard to distinguish, just think about you need to cook and then you need to digest. Okay? Cooking is denaturation and digestion is hydrolysis.